right, so this is your very first mini lecture. It's going to cover biological membranes, which is sections 9.8 and 9.9 in your biochemistry book. So hopefully you've already kind of read through those, or at least actively read through those before watching this lecture. All right, so what do cell membranes do? That would be a good first question to ask yourself if you're about to learn about something. Well, the one thing that they do that is most important is they make life possible. Think about a cell and all the different things inside the cell, the different metabolic reactions and pathways and organelles that all function in unison to give that cell life. If that didn't have a membrane around it to enclose that in a very close quarters, then you would just have a big soup of molecules and organelles just floating around, and life might happen, but maybe not as fast. Like, eventually the chemistry would occur, but not as quickly. So the formation of lipids really allowed life to boom and move a lot faster because you're holding everything really close in close quarters. So things can happen a lot quicker. Cell membranes provide protection for the cell, for the internal workings of the cell. They can also allow for compartmentalization. So think about most organelles have a lipid membrane that holds all of the functions and different enzymes within that organelle in that space. Having a membranes can allow for different concentrations on either side of the membrane, and you'll find that in many biochemistry reactions that a concentration gradient can be a really great driving force for a reaction to move forward. Membranes also act as receptors. There are proteins that are within our membranes that interact with signaling molecules that are going throughout the environment or within your body, like different hormones. And so membranes act as receptors to those molecules. Membranes also allow us to transport nutrients inside, bring in things like sugar or nutrients, different cofactors inside the cell, and also allow us to get rid of some waste, so excrete waste outside of the cell. And lastly, the membranes are kind of like an identification, like an ID card for different cell types. So depending upon what types of carbohydrates are typically displayed on the outer membrane of a cell type, that usually can indicate what type of cell you're looking at. So they can act as signals to other cells, the membranes can, based on what's on the outside membrane. Okay, what are cell membranes made up of? Well, three major macromolecules. That's going to be proteins, lipids, and carbohydrates. And probably before maybe you see this chart I'm about to look over, if you were like me, you'd think, oh, I bet it's like 80% lipids, right? We always talk about the lipid membrane. But in actuality, when you kind of look at some of these different percentages of different cell types, it's pretty impressive that almost 50% to 75% of cell membrane composition is actually proteins. So proteins, again, those are going to help us with our signaling. Those are going to help import nutrients, export wastes. They just play such an important role that they're definitely undershadowed. I mean, it shouldn't be called a lipid membrane. Maybe it should be the protein membrane. But again, so lipids typically from this chart looks like they make up anywhere from 20 to 50, a little over 50% of the membranes. It just depends on what cell type you're looking at. And then lastly, carbohydrates. So carbohydrates make up a very small percentage, from about 0 to 10%. And those are always going to be decorated, the carbohydrates on the outside of the membranes. You'll never see carbohydrates facing inside of a cell membrane. They're always going to be facing outside, displayed on the outside of a cell. Okay, so what do cell membranes look like? Well, we have a few terms to go over here. So that the one that you probably are most familiar with would be it's a lipid bilayer. Lipid because it's made up of lipid molecules, and bilayer because it forms two layers, and those two layers have names. The outer leaflet is the leaflet that faces the external environment, and the inner leaflet is the one that faces the cytoplasm, or the internal part of the cell. So you've got your outer and your inner leaflets. How can you describe the structure of a membrane? Well, there is this term, fluid mosaic model. That's the big one that's been coined. So fluid because everything within the membrane is constantly moving. Mosaic because it's made up of many different things. So a mosaic piece of art is some piece of art that has different things. And in this case, this is a mosaic of different tiles, different colored tiles. And then model because it's a model. So it's a fluid mosaic model. So fluid, it's always moving. Mosaic, it's the three major macromolecules make it up, and then it's a model. How do we know it's fluid though? Well, there was this excellent experiment done where they labeled with green fluorescent protein some proteins on a mouse cell, and they labeled those same similar proteins in a human cell, but only with red fluorescent protein. So if you take those two separate proteins and you fuse them together and make them one giant cell, you see, or in the experiment, they saw that over time, and in about 40 minutes time when they went back to check, that there was an equal distribution of red fluorescent protein and green fluorescent protein, which would indicate that, right, everything's fluidly moving around. If it wasn't moving around and it was all rigid and solid, then after 40 minutes, you would still see separation of your green and red fluorescent tags. But again, it was all evenly mixed around. So this is how we know it's fluid in the fluid mosaic model. All right, this is a very short video that really summarizes a lot of those points. It's great. So it's really old too, which makes it fun.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
So I really hope you're able to hear that, because I think I might have to click a function, but I'll see when I play it back. If not, I'm going to put this video, post it up right under this one on Canvas in the notes, so you can see it there. But again, it's just a really good summary of everything I pretty much just said. Okay, so moving forward. Here we go. All right, next we're going to talk about specifically the different types of lipids that make up lipid membranes. So the basic structure of a lipid that's in the membrane is you're going to have a hydrophobic tail or tails. That could be 2. Typically, it's either 1 or 2. And then you have a hydrophilic or a water-loving polar head group. So there are 3 major types of lipids that make up all lipid membranes, and that's going to be phospholipids, sphingomyelins, and cholesterol. So I'm going to go over the structures of each, and we're going to show where's the polar head group and where are those hydrophobic fatty acid tails, or where's the hydrophobic part of the molecule. So for phospholipids are made up of a glycerol backbone. Sometimes these are also called glycerophospholipids. And so glycerol, you have to figure out what is glycerol. Glycerol is a small 3-carbon chain that has alcohols off of each carbon. So those alcohols are now hidden into other functional groups because it's been, assembled with things, but it's the smallest carbohydrate. It's 3 carbons long and each carbon has an alcohol off of it. That's glycerol. So in this phospholipid, off of one of the carbons, you'll see there is a phosphate group. And typically there's more than just phosphate. There's an additional something attached onto that phosphate. And some examples are choline, serine, ethanolamine, or carbohydrates can be attached on there to that phosphate. Okay, but off of 2 carbons in your glycerol chain, you'll have the fatty acid, free fatty acids attached on there. So a free fatty acid would have had a carboxylic acid right here, but that carboxylic acid becomes condensed with the alcohol and forms this ether bond. This ester bond, E-S-T-E-R, the ester bond, and then also releases a water molecule in the process. Okay, so that's how you condense on the fatty acids to the glycerol chain. And then, so that's kind of the general structure of a phospholipid. The general structure of a sphingomyelin, it looks similar, like it has two long fatty acid tails, kind of like the phospholipid does. But if you actually look and analyze the structure, you can immediately spot a couple differences. So these two fatty acids both have the ester bonds, but over here, this fatty acid is linked up by an amide bond, a C double bond O N. So you see an amide, so there's a nitrogen group. Sometimes in these sphingomyelins, this is even just an NH3 by itself. So it might not even have a fatty acid off of it. But so the nitrogen kind of off of this little glycerol-like area of the molecule, like a 3-carbon-ish unit right here. So we have the amide bond. And then the other thing that's very different is you'll see there's a free alcohol. And just below that free alcohol, you should find an alkene, a double bond, and then after that you see the long fatty acid tail. So the big differences are, look for an amide bond or a free amine, a primary amine here, and then secondly, one of the fatty acids will have an alkene in it, and just like absolutely no ester bond whatsoever. Our phospholipid, always 2 ester bonds. Over here, no ester bonds. There's either an amide and or this kind of alkene area. And later we'll see the synthesis of these molecules. But for now, you just need to identify differences. And then lastly, cholesterol. Cholesterol is the last lipid that makes up membranes. It has a tiny polar head group, which is this alcohol, this oxygen right here, kind of what makes it so polar. And it has this part, which is the hydrophobic region. I was going to say hydrocarbon, but I guess it is mostly hydrogens and carbons, isn't it? Cholesterol is a steroid molecule. You can tell it's a steroid because it has this really characteristic 4-ring fused system, which would be 3 6-membered rings followed by a 5-membered ring fused onto that. So that's a very bing, like you see that little ring system pattern right there, and you immediately need to think that's cholesterol or some type of steroid molecule. All right, so those are the 3 lipids that make up the membranes. And we'll talk a little bit about how they move around in the membranes. Remember, it's a fluid model. So lateral diffusion, which would be within the same leaflet, either the outer or the inner, that occurs rapidly, very, very fast. I think it's even like the lipids switch places with the partner right next to them over 1 million times a second. So within 1 second, they have flipped places 1 million times. So lateral diffusion is incredibly fast. You also, though, can have transverse diffusion. So that's moving from one leaflet to the other. So like flipping in or flopping out, kind of. In order to do that, though, you have to have an enzyme, because you're not going to be able to drag that polar head group through that super hydrophobic region and then like turn it 180 degrees and then shove it down, right? So you're going to have an enzyme that's going to be able to grab that and flip it for you to protect it as it's moving through the hydrophobic region, which there are 3 types of enzymes. They have a pretty fun naming system, I think, too. They're flipases, they're flopases, and then there's something called scramblases. So flipases and flopases both require the use of ATP to do that, to move the lipids. And then scramblases do not require energy, and they just can move outer to inner or inner to outer leaflet. They can do both. So flipases flip things in, so they move from the outer to the inner membrane. Flopases move things from the inner to the outer membrane, so they'll move in to out, and again require ATP. And then scramblases do not require any energy, and those can just flip the outer to the inner and the inner to the outer. The actual composition of the outer and inner leaflets are very different. Again, carbohydrates are only displayed on the outer leaflet, yet they are synthesized usually in the cytoplasmic region. So you're going to make these lipid molecules that have this kind of, ID cards on them based on the different types of carbohydrates. And then one of these enzymes, likely the flopase, is going to identify that and then flop it to the outer leaflet. So these enzymes, these 3 types of enzymes, play a very important role in maintaining the lipid leaflets. Okay, so let's talk about, it's enough about the lipids, let's talk about the membrane proteins. So membrane proteins, there are 3 types. There are integral membrane proteins, which the easiest way to spot those is they're going to span both the outer and inner membrane. There are peripheral membrane proteins, and those are typically found only against the inner leaflet, and they're just kind of hugging and interacting with the polar head groups. They're not covalently attached, it's only via non-covalent interactions are they hanging out with that inner leaflet. And then you have lipid anchored proteins, which are proteins that somehow have some type of a lipid molecule that anchor them into the membrane space. So let's look at these in depth. All right, so like I said, integral membrane proteins are going to span both the inner and outer leaflets. It's going to create, they usually typically create pores. So this is where nutrients can come in, right? How are you going to get a big polar glucose molecule from outside the cell inside the cell? Well, it's going to have a help of one of these proteins that makes a nice pore for the glucose to come through. So they aid in membrane transport. And the other thing that integral membrane proteins aid in is the signaling. These typically are your receptors. Signaling receptors are membrane proteins that are going to be integral membrane proteins because there usually is likely an extra peripheral protein right here that's interacting with the inner side part. So once a signal binds the outside, creates a little conformational change that'll kick off the peripheral membrane protein, and then it can go and start cascading that signal to get the cell to do the new thing it needs to do. Peripheral membrane proteins, like I said, again, are on the inner leaflet, and they are usually being held together through ionic and not all the non-covalent interactions with the polar head groups. You can easily remove a peripheral membrane protein if you change the pH or maybe change the salt concentration around. You can kick those off pretty easily. Just depends on the types of non-covalent interactions that are holding it there in the first place. And lastly, the lipid anchored proteins. So there are 3 types of lipid anchors you can find. Fatty acids, perennial, which is also isoprene, and then the phospholipids. Essentially, I would just call it like a phospholipid. That will be easier to remember. So here's an image that shows those 3. So the fatty acids are the easiest because here's our protein, that's a protein here that is just tethered to one single fatty acid. So this, again, a free fatty acid, that would have been a carboxylic acid, but it's going to be condensed to an amino acid on a protein, one that has an alcohol, which there are 2 amino acids with alcohol side chain functional groups I can think of. Can you think of them? Hopefully serine and threonine, those came to mind. Yeah. So you're going to covalently affix a free fatty acid onto a serine or threonine, and then that's how you have that lipid molecule that's anchoring now that protein in the membrane. So the isoprene or isoprenes are 5 carbon, it's a small molecule that contains 5 carbons with this kind of repeating patterns, a few double bonds in it, and they can readily polymerize and make these lots of different flavor compounds or different volatile, very like fragrant compounds. Plants make a lot of different isoprenoid compounds. Cholesterol is actually synthesized from isoprene. Okay, so there's another little piece of that puzzle for you. But here's kind of what the isoprenoid or the isoprene lipid anchor might look like, and it's attached to a sulfur. Which amino acid has a methylene and then a thiol, an SH at the end? Cysteine, yay, you got it right. So cysteine would be how you would covalently attach the isoprene units onto there. And then lastly, you have your glycerophospholipid. So here you can see it's got the glycerol, and you have your 2 free fatty acids. There's the phosphate. And then there's a bunch of different sugar molecules. These are just different sugar molecules. N-acetyl is this sugar specifically, but there's some other ones there. And then it's linked through this ethanolamine residue here. But again, there's that protein. So I would just call it kind of the like phospholipid anchored protein. So free fatty acids, the perennial or isoprene, and then the phospholipids. All right, so what are the big takeaways from this mini lecture? You should be able to provide definitions for the following. You should be able to define what is a lipid bilayer, the inner and outer leaflets, hydrophobic, hydrophilic, the fluid mosaic model, the 3 different types of proteins, integral, peripheral, and lipid anchored, the three different types of lipid molecules that make up membranes: phospholipids, sphingomyelins, and cholesterol, as well as the three enzymes that help flip those lipids between leaflets: the flipases and flopases, and the scramblase. You should be able to label the polar and nonpolar regions on phospholipids, sphingomyelins, and cholesterols. And you should be able to draw and label a detailed cell membrane that includes the three different types of proteins, so all labeled, the three different types of lipids, and again, probably a rough sketch. Like, don't worry about getting exactly the right number of carbons and things. Just remember all those little indicators I was saying, like how you know one's a sphingomyelin and one's a phospholipid, as well as where is the location of the carbohydrates. So remember, carbohydrates are only ever on the outer leaflets. They're not on the inner leaflets. So they're being, expressed to the outside of the cell, to the environment.